Okay, so the 44 multiple choice questions. So when you're looking at this material, I'll definitely be asking about what the newest uh, ratio is, because there's so much out there with the wrong ratio. I'd like you to really get that memorized and what the new ratio is. So it's closer to almost 50-50. It's 1.3 to 1. So it's a little bit more microbes than human cells. So you can still say you're mostly microbe, but it's not as, some of the older videos and older readings, they are mistakenly saying, 90% of your body is not you. So just make sure you know the true estimate. And then we got into a little bit with what happens, when you're either in the uterus. So as you're studying this, I will be asking questions about these ideas, what's going on with the uterus environment and what happens with the mom, as she's carrying you with either her diet or breastfeeding, formula feeding. So we covered that material in this first slide set. So make sure you're reading through these and thinking about it and what's similar, what's different with C-section versus vaginal birth, similar, different with breastfed formula. So make sure that you're looking at these and kind of thinking about key ideas and kind of comparing. And then this slide set kind of gets at, okay, once you're born, then what? And then it gets into some of the different parts of your body in terms of who's there, how many in general are there. Do you have any questions on this slide set? Yes. No, I won't say, what's a common microbe in your throat? I won't do that. No. But looking at the entire human, what's going on in the upper versus lower. And then we got into the intestinal tract and looking at, this progression of from few microbes in your stomach to way more down in your colon, so these types of ideas, but not specific species. Mucus. Make sure you learn about mucus. Very important. All right. So this is kind of a really key concept that they're starting to hone in on, as if you don't have good mucus design in your intestinal tract, things can go wrong pretty quickly. So learn about the mucus through the GI tract and what's going on when it's nice and healthy versus how does it get into a perturbed and degraded state. And then inflammation is huge. Kind of goes hand in hand with a degraded mucus. You tend to get this inflammation issue. But remember, when they say inflammation, they generally mean body-wide. They're not just, it's not like when you get a paper cut or something on your finger where the inflammation is just right there. They're pretty much saying this can be a systemic body-wide issue. So thinking about these ideas, because it's just, where is that? This slide number 60, this scientist is saying pretty much everything is coming down to chronic inflammation in terms of the different diseases that are out there. And that current medication is really only trying to treat the symptoms, not the actual cause, which is, of course, what you want. You want the actual cause to be fixed. Questions on this slide set? I'll go through the different body parts and ask you like the multiple choice questions on um, the different body parts in terms of what's going on there with regard to the microbes. And then mucus, that's a pretty big idea. So I wouldn't be surprised if you would see an essay question on that because that's a pretty important concept. I want you to demonstrate you are really grasping what's going on there. And the best way to do that is through short answer or essay. Second slide set, part 2. So yeah, I definitely will be asking you like, how do they do this? Because I think it's important that you understand how they're getting this data and that it's not perfect data. You know, a lot of it is, it really does depend on how deep do they sequence, how are they sampling. You know, when I listened to Jessica's presentation, when she gave her PhD defense, you know, she was trying to get at what's in the mucus. And she's, you know, came to the conclusion, feces, those fecal samples don't get you there. If you really want to know what's going on in the mucus, that's not the best option. That's where the engineered pill is coming into play. So hopefully they'll get that, someone will get that designed and out there. But this slide set's looking at, you know, sampling, experimental design, processing. So I'll certainly ask about that. And then again, we're getting into other parts of the body, what happens with your brain. So how the gut brain can influence the brain in your head. So think about those dynamics. So this kind of is looking at how microbes in the gut can actually influence distant parts of your body, including how you think and feel, anxiety, depression, and others. And then we got into some of the jargon with psychobiotics, probiotics, prebiotics, synbiotics. So of course I'll be asking about those. These slides here, 41, 42, 43, 44, 45, those are important. So this study, where they did the identical twins, be able to talk about that. I want you to learn it well enough where you can give me some, so that's probably going to be an essay, okay? But those are important concepts. And there's, of course, short chain fatty acids. We're looking at that idea with the microbes producing those three. So those three fatty acids are like 98% of the fatty acids that microbes produce for you. It's those three. And they're finding that they can have profound impacts on you. And you can alter that by what you eat primarily and your general lifestyle, including exercise. I won't ask you specific on like diets. Like I won't say, you know, tell me what's happening with ketogenic diet. I won't do that per se, but looking through those slides and thinking about, you know, what looks to be something that would be more optimal, what looks to be something not so great, and why. Like these slides are visual, but they're showing you what's going on that's good and what's going on that's not so good. So looking at those, but I'm not asking you to memorize the different diets and spit out information on that. Questions? What other questions do you have on human microbe? Any questions? The postbiotics?
Yeah, how are they identifying those to then be able to sell?
I think a lot of it is they sample human blood, because a lot of postbiotics are in your blood. And that's probably the easiest way to start screening for them. So like, one of the postbiotics too is like the short chain fatty acid. So, I mean, I think they are currently selling like butyrate supplements that, you know, you can take. But I think when they realized how much is coursing through our blood system, that's where they started. I don't know how specific they get, like, are they, you know, are they doing studies with the gut microbiome specifically for this? I would assume they are. And a lot of what they're doing is kind of the transcriptomics, where they're looking at what messenger RNA is being expressed. And then they can tie that to what protein is that and what's going on there. So I think it's a combination of looking at the messenger RNA. I saw another hand somewhere. Oh, yeah.
Is there any questions from the TED Talk?
The Paul Ewald TED Talk, the homework assignment? No. Nope. Nothing on that. You already did that for the homework. What about, I'll leave this open in case you come up with another question, but let's go into food. Food microbiology. I'm really not going to ask you about details of food spoilage. Just know what food spoilage is. It's basically when they microbes create a situation that you find distasteful, but it's not pathogenic. It's not like a pathogen that you ingest. So food spoilage is one thing, and microbes are certainly responsible for a lot of that. But then you get into, so I won't ask you like details on off the tables or anything for the food spoilage. So then we get into like water activity and, you know, where are these microbes lurking that we may need to worry about? And, you know, what if pathogens are lurking in some of these places? And then I just gave you a couple of examples of how, you know, with our current way of doing the food industry, including how we raise and grow food, how we process it, how everything's kind of consolidated. So think about those ideas. And then we looked at poisoning versus infections. So food poisoning versus more of a foodborne infection. There's a clear difference, including symptoms. And then we got into endo and exotoxins and also enterotoxins. So that table found on slide 37. Make sure you spend some time with that as well. Questions on food micro?
Remember specific species?
Probably the ones listed on slide 29. That would be good. Salmonella, Campylobacter, E. coli, Listeria. And then probably the Staph aureus too. Slide whatever that is. What is that?
And
34.
would you like us to know just like what foods they would be in or like what conditions they grow in? Or
Kind of conditions they grow in, you know. I might throw food in there as an extra tip off, but kind of looking at, you know, we talked about like Staph aureus. I mean, Staph aureus shows up in so many different things. But when you get sick with something like Staph aureus, that's pumping out toxins, you tend to feel it right away. It doesn't wait. You start feeling pretty yucky within a matter of hours.
To know the reaction we have to that specific or that specific.
No, like symptoms? No, I won't ask symptoms. Other than timing. I think the timing is important, you know, like how fast does it hit you? I tend to use that when my boys get sick. It's like, okay, what did you eat? When did you eat it? And then I can try to figure out what's going on. Other questions on this food micro?
Distinction between the infection versus the
Poisoning? Slide 34 talks about poisoning where it is a preformed toxin. We're talking about you eat food, let's say maybe it's a donut with that custard filling, which I love. But anyways, so Staph aureus can lurk in the custard. And they are already pumping out toxins as they're in there, as they're having fun in the custard. They already are pumping out the toxins. And so as soon as you ingest it, you're getting exposed. Whereas food infection, it's more of you ingest the bacteria in the food. And then it has to get to a high enough population before you start feeling not so good. And it can get pretty bad, but it tends to kick in, you know, like eight hours or longer. Other questions?
Specific to this slide set, but how many essay questions are you gonna have?
Essay, I think, is 5 right now. I have a pretty solid draft. And I have 5 essay and 1 definition on that section of the test. Applied and industrial, which we just wrapped up, but make sure you do think about these ideas. Oh, table 16.4. So slides 10, 11, 12. I'm going to ask you, give me an example. I'll just tell you that right now. I'm going to say in table 16.4, give me an example. And the reason I want you to know something off of here is because I want you to be able to talk about how much better it is now that they're using microbes. Like, what's the benefit? So make sure you learn that too, compared to doing it the old way, to be able to give me an example off of that. And then just think about all the different things we covered. And, you know, this certainly isn't complete. Microbes do a whole lot of other stuff. This is just things I could tell you in our short time together. So make sure you are looking through these. And I'll certainly be asking questions on synthetic biology, biotechnology. Questions on stuff in this slide set? Okay, what about the cumulative? So I posted that Word document on what I'm going to be zooming into. Yes, no. Nope, I don't think anything is other than the definition of a prophage. Like, that's all I'm gonna ask. What is it? And then the rest of the essay is new stuff. So there will be some cumulative questions based on that Word document. Which I don't have up in front of me. So do you have, does anyone have questions on those concepts? Yes.
You just like refresh my memory on oxygenic versus oxygenic photosynthesis.
Yes. Or not photosynthesis, and just metabolism. In fact, I don't think there's a single photosynthesis question on this one, so. But let me pull up that slide set. Let's see. Where is it? Not that one. So oxygenic versus anoxygenic metabolism. What did I say? No, I'm thinking of the wrong thing. Oxygenic. Is that what you asked?
Yeah. Oxygenic.
Oh, all right. I was totally thinking of something else. All right. So oxygenic. So sorry, let me rephrase it. This is like the only thing getting into photosynthesis. I was thinking about anaerobic metabolism when I answered your question. So this is what? Is this oxygenic photosynthesis or anoxygenic? It's oxygenic. What does oxygenic even mean? Creates oxygen. So if something is creating oxygen, that means it's a product. And the only way you can do that in photosynthesis is if you use water as your donor, your electron donor. So that's really what you're trying to piece together. It's like, okay, oxygen's a product. It's oxygenic. Therefore, water must be getting stripped of electrons because that's how you get oxygen. So they must be using water. And we know like cyanobacteria, they do that. They strip electrons off of water. And oxygenic, there's a couple of different ways microbes can do that, but I'm not going to get into the details of the different ways. But thinking about anoxygenic, where is it? I guess. Let's go here. So this is anoxygenic. Why? Yeah, it's using hydrogen sulfide. You can't get oxygen off of that. That's not one of the products. So it's using something other than water. What was some of the other questions on that Word document? The aerobic versus anaerobic, was that one of them?
Yeah, the respiration.
Yeah, okay, let's go to the, whoops, go to that one. All right, so I noticed on one of what was it, the second exam, when I had a picture and I said, what's going on here? A lot of you picked, oh. So I had on, I think it was exam 2, I had a picture of this. What's showing up in figure B, when they said, tell me what's going on here. And the correct answer was anaerobic metabolism. But a lot of you picked denitrification, which this is not denitrification. That's not denitrification. Denitrification would be if you take nitrate and push it all the way to dinitrogen gas. So this is just one little step here. This is not denitrification. But it is anaerobic metabolism. So when you hear aerobic metabolism versus anaerobic, this is what you're thinking about. You have the same idea of an electron transport chain process linked to proton pumping to get you a proton gradient. It's just, what do they stick at the end? Do they put oxygen at the end as the terminal electron acceptor or something else? It's not oxygen. And very often nitrate's a common one, but not always. Pick other. So this is the difference. What were some of the other questions on there?
That's electron acceptor is best electron donor.
So what is the best electron acceptor? Oxygen. Oxygen is the best. So then, if you're thinking about, well, what would be a really good donor? We're back to this idea. So this redox tower is setting up where the very best or really good electron acceptors are at the bottom of the tower. So that's why you see oxygen is right at the bottom. Because if you think about this tower and you think about electrons are falling, so to speak, the farther they fall, the more bang for your buck, the more ATP you can get from this. And so if you're looking for a good electron donor, you have to go to the top, or the top of the tower, right? And so, and the way this works, remember, whatever's to the right of the slash, that's the partner that can donate electrons. So glucose, this HSO3 minus is at the very top, so that's a really good donor. Glucose, hydrogen gas, even methanol. Okay, so those are good donors. And we did talk about hydrogen gas a fair bit. Because that's how chemolithotrophs often will look for that. Because it's pretty abundant where they like to hang out. Other questions on the cumulative? I can't remember what the other questions were off the top.
Ectomycorrhizal and BAM function.
Oh, the fungi. Okay, fungi. So ectomycorrhizal, endomycorrhizal. So knowing the basic, so study that using compare and contrast. Fungi. So this won't be an essay question, but think about comparing and contrasting. How are they similar? How are they different with how they interact with the plant roots? By the way, the question on the last exam I had asked, I showed you a picture of this one right here. So the roots with the mantle on the short, fungal mantle on the short roots, and then the hyphae coming off the root. A lot of you missed that one, but that's that fungal mantle idea. That's typical of the ecto with that Y shape in particular, very typical of the ectos.
So that's growing off of it.
So this tan, the tan part is the root, and then all the white is the fungal tissue, fungal hyphae. Questions on other aspects of the cumulative? Yeah.
Fungi, I think, I don't think, fungi, we did, endomycorrhizal. Wow. What's the class of fungi that goes in endo and extra?
So the glomeromycetes are the endos. The endos belong into that one. No. Ectos are usually the basidiomycetes. You find the ectos in that group, that class. Oh, how many questions? I think I asked one question. How many, how many, topics do I have on the cumulative? 6, so probably about a dozen points. Probably about that. Other questions? How many points? For sure, the biggest point value is human microbe. And then I'd rank it next would be industrial. And then food.
Do the point breakdown like we did last time.
Send it, yeah, when I finalize the exam, I'm pretty close to finalizing it, so I can give you a point breakdown. Yeah, so that's Monday at 2 in this room, okay? Same idea, multiple choice, separate essay, okay? So come prepared with your laptop charged up and good to go. I will be checking my email over the weekend, so if you have any last minute questions, make sure to send me an email, okay? Hang in there. We're getting close.